Okay, hi. Since there were some power outages and we're not having class on Monday, I've briefly narrated this PowerPoint just to give you some information beyond Mendel of some complex inheritance patterns. Again, most of what we've talked about is Mendelian genetics, but this will give you a little insight about some of the things that we've learned since Mendel. I've outlined some questions here. Specifically, we're going to speak about what do we mean by gene interaction, what is epistasis, what are discrete traits or versus quantitative traits, and how do we know genes do not always assort independently, and what does that mean? And that means we'll talk about linked genes very briefly. And then lastly, what is extracellular inheritance, and specifically, what is maternal inheritance? Okay, so just again, a reminder that, sorry, I'm trying to use the pointer. Just a reminder that, oops, sorry. Yep. Okay. Just a reminder that, again, so in reality, most traits are influenced by more than one gene. So for example, one's height. Many genes are involved in that. Many proteins are involved in that. And there's variation in that. And we will see that in a few minutes. Whereas Mendel thought of one gene, one trait, a single locus, which is a site on the chromosome, one trait. However, again, the take-home message where the yellow star is that what we see more like true is gene interaction, where one trait is controlled by more than one gene, two or more genes. Okay. What we're looking at here is a very important phenomenon we call epistasis. So again, this has to do with the fact that you have two genes involved in a trait. And in epistasis, or epistatic gene interactions, the alleles of one gene mask or cover up the expression of another gene. And we're going to show you this experiment by Bateson and Punnett, sweet pea crosses, that illustrates this. So what they did, and knowing what you did about Mendelian inheritance, you could assume that to be true first. So for example, they took a true breeding purple sweet pea plant and they crossed it with a true breeding white flowered sweet pea, and they got all purple. So it seems like from Mendelian genetics, you have dominant purple crossing with recessive white, and at the first generation, F1, all are purple, right? And then they did another cross of the F1, and if we use what we learned from Mendel, in the F2, you would probably get a 3-to-1 ratio, 3-to-1, where you have 3/4 were the dominant, in this case purple, 1/4 were the recessive, in this case the white. Instead, they so they did get the 3-to-1, so that was fine. So then they said, well, let's take the white and cross just white rather than white and purple. Let's just cross white with white. Now, if you had a recessive situation, you cross white with white, what would you get? White, right? However, they got a big surprise. So here is the situation. They decided to just cross the recessives, they thought, white with white, and they expected white, and they got purple. Weird. And then they crossed the purple, and then they got some weird outcome. So they definitely did not follow Mendelian genetics. So what they determined was that there was epistasis going on. And so what they determined is that for this particular color flower that there were two genes involved to make something purple, the flower's purple. So there was a gene with the letter C and a gene with the letter P. And so those two genes were involved in some pathway, which we'll look down here. And so they determined that you need to be dominant, or both to be dominant, to get purple. That means that you need to have at least one big C and one big P to be purple. So for example, if you're big C, little p, you won't be purple. Or if you're little c, big P, you won't be purple. Or the heterozygous situation with the recessive, you won't be purple. You need to have at least one C and at least one big P. So one big P and one big C to be purple. And that's what's determined. So the alleles of one trait, the alleles of one gene mask the alleles of the other. So even if you have, the big P, these alleles, the C's, will mask that. And so this organism will not be purple. So in order to be purple, you need to have one dominant C and one dominant P. And so they've determined that two or more genes are involved in this pathway. So if you think of it this way, let's say you have a colorless precursor, and then this enzyme C, which has this gene C, you need a capital C, big C, to then take you to this intermediate, and then a capital P to take you into that last step, which is the purple pigment. So you need both of those to each have a dominant allele. So that's called epistasis. Okay, a little bit more about multiple genes. So again, Mendel talked about discrete traits, um, pea plant flower color, something specific. One gene affected that. However, what we've seen more of is what we call quantitative traits, where the traits are determined by multiple genes. We call it polygenic, meaning many genes. So quantitative traits contain many genes, or they're polygenic. And the other thing about quantitative traits, besides containing many genes, the traits themselves have a range of phenotypes. So for example, height. People of different heights, right? Not everybody is either 5 foot or 6 foot, right? Like there's a whole range of phenotypes of height. So that is considered quantitative trait. There's a continuous variation over a range of phenotypes. It's made up of it's a polygenic experience where you have many genes involved, and also environment can actually play a role. And so what we'll see in this example. So in this example, we're looking at a polygenic or quantitative trait of skin color. I don't know if you can see, but there's multiple alleles, or multiple genes, actually 3, A, B, and C, if you can see all these. Those are all involved in skin color, and what's plotted here is the different genotypes and how that affects the color and the fraction of people with the same phenotype and things like that. And so they're looking at like the range, and there's a big range here of phenotypes, as you can see. So it's not just one box or one column. It's a whole curve here. And again, that will look the assortment of those alleles. Again, there's 3 genes involved. Will determine a color of skin, but not everybody has the same color or shade or things like that. So that's considered a polygenic or a quantitative trait. The other thing is that the environment can change the skin color, as you know. You've been out in the sun. So you can see a different shift of this curve based on if the same population was raised in a sunnier environment. So again, and this is pigmentation from light to dark. Okay, so this is a quantitative trait. I'm just going to mention this. I'm not going to really necessarily ask you any extensive questions about this, but it is good to know that in a female mammal, one of the X's they have two X chromosomes in the female. One of the two X's in each cell of a mammal, female, is randomly inactivated during the embryonic development. So there's one, basically after this is all done, there's one active X and one inactive X in each cell. So whatever is the active X will be what they see, what you see, the kind of gene. And so, for example, if a female is heterozygous for a particular gene located on the X chromosome, then she'll be what they call a mosaic for that character, because in some cells one of the X's will be inactivated and in other cells the other X will be inactivated. And so this is best seen on a tortoiseshell cat fur. Okay, so you can actually see in the case of the fur here that like for example in this kind of cell, this is the inactive X, the one with the black allele, and here in this case this is the inactive X with the orange allele, so this will appear black where this will appear orange. So the animal will actually, the fur pattern will be like a mosaic. It won't be any consistent color. It'll be a mixture, alternating random type of mosaic. So that's called X inactivation. And it only occurs in females because females have the two X. Okay. We're just going to enter a little bit of a discussion about linked genes. So we learned with Mendel that, like, the second law of Mendel was that the law of independent assortment, where alleles of different genes or different traits assort independently. They're not sticking together. They're assorting randomly, independently during meiosis when the gametes are formed. That was what Mendel thought. But since Mendel's findings, others after him have learned that, well, inheritance doesn't always work this way. Genes can be on the same chromosome, which then means they'll tend to be inherited together if they're close together. And we can actually use the inheritance of certain genes that are close together to figure out like how far apart they are. And you can actually map chromosomes based on the inheritance of alleles that are close together on chromosomes. And so Thomas Hunt Morgan did some work, and also Bateson and Punnett. So those were all geneticists at the time. And so they basically come up with the conclusion that genes do not always assort independently, especially if they're linked. Okay, so independent assortment only works for genes on different chromosomes. But if we see linkage, which means when two genes are close together on the same chromosome, they will tend to be transmitted as a unit. We call it a linkage group. So what I'm going to show you now, I'm going to simplify. I kept the information in the PowerPoint in case you'd like to go deeper and try to understand more, but I'm not going to hold you responsible for a lot of it. So I'm just going to tell you kind of what you need to know. So certainly you need to know that linked genes do not assort independently. They do not follow Mendel's law of independent assortment. So they found this with this particular sweet pea cross they did, where they had like the two dominant traits, purple and long, crossing with the two recessives, red and round. And the ultimate result, if you did like a dihybrid cross, normally we got a 9 to 3 to 3 to 1 ratio. In their case, they did not. So if you do a dihybrid, what you think you're doing, like here, you start out with the parents, and then you get the dihybrid, and then you cross the dihybrid, you would expect a 9 to 3 to 3 to 1 ratio, but you didn't get it. So if you don't get a 9 to 3 to 3 to 1 ratio for supposedly a dihybrid cross, then that must mean the genes could be linked, meaning those genes are actually on the same chromosome. So Thomas Hunt Morgan, followed up with that, doing fruit fly experiments. And again, he also found that you couldn't explain everything with Mendelian genetics. He came up with the same idea that genes on the same chromosome are inherited together. Okay, so that you need to know that, of course. If genes are on the same chromosome, you can actually get crossing over between the genes, and that can result in a new combination of alleles. And we knew that from um, our discussion in meiosis. Prophase 1, you do get crossing over, and you get new combinations of alleles on those chromatids, right? And so on the last point, I'll just mention too, is that when you do get this crossing over between alleles on a chromosome, that depends on the distance between those two alleles. So if you have a wider spread distance, you'll get more crossing over between the alleles. If the alleles are really close together, you might not get crossing over. Okay, think of if you're sitting next to a friend and someone wants to kind of cut in between your seats. It's hard to do if you're very close to those, if those two chairs are very close together, but if you split the chairs a little further apart, then people can cut between you, no problem, right? So what I'm just showing, again, I'm giving you all the information. You don't need to worry about this right now, but this is what they did. They did a series of crosses, and what they came up with is that doing these two traits in each parent, they had like a gray body straight-winged fly and a black body curved-winged fly. The majority of the offspring look like the parents. Gray body, straight wing, black body, curved wing. A small percentage we call recombinants have the opposite phenotype. So they have gray body, curved wing, black body, straight wing, and that must mean that there must have been some crossing over. So, and then through this experience crossing over, you can actually, by the amount of crossing over, you can do a mathematical equation, which is on the next slide, which again, I'm not holding you responsible for, but just in case you're interested, you can use this equation based on how many recombinants there are to figure out how far apart genes are on the chromosome. So that's how geneticists did. So for example, they've been able to map this chromosome, and they see that these are all the alleles on the chromosome, and this is where they're located. And that was helping them, those experiments that they did were helping them figure out how far apart alleles were on a chromosome. And this is that equation I mentioned where you do not have to worry about it, but if people are interested, based on how much new offspring, where the offspring looks like a mix between the two parents because of the crossing over, if you look at the percentage of how many of those unique offspring you get compared to the normal offspring, like that look like the parents, you'll get a number, and then that will tell you how far apart the alleles are on the chromosome. So again, I really just want you to understand that linked genes do not assort independently. You don't get a 9 to 3 to 1 ratio, and that, linked genes are inherited together pretty much. Okay, and the last thing, we've been talking all about nuclear inheritance, and the genes are in the nucleus. Well, we also know from studying the cell that there are extrachromosomal organelles like mitochondria and chloroplasts that also have DNA, also have genes. So we call that extra cellular inheritance, meaning mitochondria and chloroplast inheritance. And so, when during reproduction and things like that, the chloroplast and the mitochondria are found in the cytoplasm of the egg, and so because the egg gets fertilized, whatever is in the cytoplasm will be from the mother, and so because a sperm doesn't transmit any of that stuff, the sperm just transmits its nuclear DNA. So whatever cytoplasmic DNA from the mother is there, that's what's going to be coming out in the offspring. So for example, for chloroplasts, what you can see here, and you have to worry the detail, but so in the case of studying inheritance in a chloroplast, for example, these are the symbols, female and male. So if you cross a female with white leaves and a male with green leaves, the offspring will all have white leaves because they get their white leaves from the mother, because the white leaf gene is on the chloroplast, and the offspring all get the mother's chloroplast. Okay? And same thing goes here. If you have a green leaf mother and a white leaf dad, all the offspring are green. Okay? So basically, that comes from the mother. So if it's on the chloroplast, it's going to go into the egg, it's going to come. And in humans, we don't have chloroplasts, but we do have mitochondria, and there are actually genes on the mitochondria, and there are diseases linked to mitochondrial genes, and those are always inherited through the mother. So no matter what the father's genotype is, the mother will pass that on to the children. And so there's a few diseases listed here in this chart, which, so like Leber's hereditary optic neuropathy, that's a vision, loss of vision. I believe the student who actually graduated last year, who's a dancing major, who was blind, actually had this. This is what she had, because she mentioned that in the Today Show when she was on the Today Show this past week. But that's a mitochondrial gene or disease gene that gets passed on from the mother to the children. So anyway, that is the last bit to say. I think that's the last slide. And so again, those are some of the highlights I wanted you to know. You can check the review sheet to see what else is on there, but I pretty much emphasized right now what you need to know as we conclude the genetics chapters. However, Dr. Hayes has info on quantitative genetics and things like that, and she's posted her PowerPoints and questions to guide you through that. So anyway, Wednesday we will have class, I'm assuming, and we will have time to address any of these questions that you may have on this chapter. Okay, also you can email me with any questions. All right, have a good day. Be safe.